Thank you. Thanks for having me here. Very excited to present what we've been working on in my lab here at Georgia Tech. So I want to just take a quick, brief moment just to introduce our model organism. It's not a model organism that a lot of people have heard about. We work on the tunicates, and the tunicates are so called because they actually have a thick tunic or cover made mostly of cellulose. They're actually the only animals that are capable of making cellulose, and this allows them to sort of, stay put in attached to some kind of substrate and filter feed, showing you that, they really look like, really odd sort of marine creatures, but they happen to also be the invertebrates that are most closely related to us. They are the sister group to the vertebrates, meaning out of all the organisms out there, tunicates and vertebrates are more closely related to each other than to any other organism. So although in their adult form they look nothing like us, this is an adult tunicate of the species Ciona robusta, which is what we actually mostly study in the lab. The adult you see next to a penny for a sense of scale. Although the adult looks nothing like a vertebrate, this connection to vertebrates is more readily apparent in the larval phase. And the larval phase is when you have this more typical sort of chordate body plan. You've got a notochord, sort of a hydrostatic skeleton running down the middle with some muscles on either side, and these muscles propel the larva forward in a swimming motion. The tunicate life cycle is thus therefore biphasic. It alternates between a sessile or a motile fixed adult and a swimming or motile larval phase. This larval phase is really, really short-lived. Actually, these larvae only swim around for around 24 hours. That's because they don't feed, they don't reproduce or anything. In fact, their digestive system is not yet differentiated. Their only, task is to disperse the species. This is. Thank you. This is the dispersal phase of the tunicate life cycle, right? These adults can't really move anywhere, and so in order for the species to spread far and wide, it needs a motile phase, and this is carried out by this swimming larval phase. Now, what's interesting about this larval, the larval phase is that this actually has a very small nervous system. It's one of the smallest nervous systems ever described, and it's actually been completely mapped at the synaptic level by serial electron microscopy section. So this is a sort of a diagram of this small larval nervous system in the species that we study. One of the species closely related to what we study, Ciona intestinalis. And here, this is a matrix showing you the Ciona connectome. This is just showing you the depth of synaptic contact between all the neurons to all the neurons here. So you've got all the neurons on one axis, and this is just showing you the amount of synapses and the extent of the synaptic contact between each and every neuron in this minimal nervous system. There's about 177 neurons in the central nervous system, and the rest is sort of scattered as sort of sensory neurons in the periphery. Now, despite this very small nervous system, this really minimal nervous system, this nervous system develops very much like our own nervous system. This is a chordate, just like us. So it develops, its central nervous system develops pretty similarly to how you would see a mammalian or human nervous system develop. You've got a dorsal neural plate. That's when neural induction happens. Now you've got a process of neurulation. This neural plate will roll up into a dorsal hollow neural tube, and then that will be patterned along the anterior posterior axis, giving rise to these different compartments of the central nervous system that seem to have homologs, or at least very closely similar, functionally similar compartments. You've got forebrain, you've got brainstem and spinal cord. You've got very similar cell types, neuronal types, in each of these compartments that seem to have direct counterparts in the vertebrate nervous system. So it's been, we focus on it as a model for nervous system development. It's always been, the tunicates have always been a very important developmental biology model organism. More recently, it's been sort of touted as one that's very, very good for molecular biology studies because you have the ability, probably unique ability, to be able to transfect hundreds, if not thousands, of synchronized single-cell embryos with the push of a button. So you can do things like take some sort of DNA, like a regulatory DNA, put it in a plasmid, say up front of green fluorescent protein. You just simply mix this DNA with these synchronized eggs. These eggs have been fertilized in vitro, so you've got thousands of synchronized embryos. You just push a button and they all take up this DNA. This makes it very, very, very easy to do things like label cells with GFP, so you can see where they're going. So lineage tracing or following the cell's movements. You can tag, obviously, proteins to see what's the subcellular localization of those proteins in the animal. Nowadays, you can do CRISPR very easily, right? The CRISPR sort of revolution allows you to target specific genes, knock those out, see what the effects of those that knockout is. And more recently, even people have started to do things like genetically encoded calcium indicator visualization in behaving larvae. Even optogenetics, our own principles of neuroscience course here at Georgia Tech, we've successfully had students carry out some very, very basic optogenetic stimulation of the nervous system using electroporated larvae. Now, just as easy as it is to electroporate a single plasmid, you can simply mix and match several different plasmids and co-electroporate them, and they'll all go into these larvae. In the example of this larvae in which 3 different cell types have been labeled with unique reporters in this sort of anterior structure that's responsible for the settlement of these larvae, photo courtesy by a postdoc in the lab, Florian. Now, those are the sort of techniques that we've been able to really use to our advantage. There are also labs out there that are really focused on now using these advantages to furthering the study of the behavior of this larva, especially now that the connectome is out, right? We've seen these connections, and they've started to understand some of these connections in the context of very simple behaviors that are modulating the swimming of this larva, right? This larva has to swim for a period of 24 hours. Its main purpose is to find a suitable place in which to settle and trigger metamorphosis into the sessile adult phase. So an example of some of these sort of circuits, simple circuits that have started being elucidated, there is a negative phototaxis behavior. So these larvae will swim away from the light, and this is thought to be because they are supposed to settle in a dark spot. So at some point, they're going to swim away from the light. At the beginning, they want to disperse, so they're going to swim towards the surface of the ocean. But at some point, they have to find a dark spot in which to settle. And so they do have this strong aversion to light. And this has been shown to go through these pathways connecting a group of photoreceptors. There's a directional component to this, right? Light coming from a specific direction will then control the swimming movements through interneurons and motor neurons in the motor ganglion, the sort of the spinal cord homolog. There's also a different behavior that is also light dependent, but not dependent on the directionality of light. This is a dimming response. A separate group of photoreceptors, if there's sort of a looming shadow over the larvae, they start to undergo this highly tortuous swimming behavior, thought to be sort of an escape response. So, if a predator is coming over this larva, its only real defense mechanism is to start to swim in unpredictable manners. These are, this is what's been hypothesized, but obviously we have yet to show, the actual, ecological importance of this. But this is what the thinking is. And there are also other circuits that seem to integrate not only light, but also the orientation of the larva. So what's been shown previously is that these larvae, when they're looking for a place to settle, first of all, they'll prefer to settle in dark spots versus light. So if they look at the percentage of settled larvae, they find that they prefer to settle in dark spots. But also, they prefer to actually settle on the underside of surfaces, not on the bottom, or the sides of dishes. So this is showing you that a larger percentage of these settled larvae tend to settle on the sort of the top of this sort of chamber where they're going to settle. And more recently, people have started to work out these connections, showing that there is an integration between the sort of the otolith. This is a sort of a gravity sensing organ that will detect the position, and there's an interaction with these photoreceptor pathways that will aid in sort of this behavior for the preference of settling on the dark underside of surfaces, right? So it's not just settling in a dark spot, but specifically in a shaded, secluded sort of undersurface, a boat or a cliff or something like that. In our lab, though, we really are interested in the development of the nervous system, seeing how sort of genetic regulation of developmental processes help shape this nervous system. This nervous system is for all we can tell, is invariant in the sense that it's sort of like C. elegans, right? You've got a set number of cells, and they connect in very specific ways, and so this allows you to really be able to focus in on specific developmental processes that happen over and over again every time in the same larva, and really sort of understand what are sort of the genes that are responsible for a particular developmental process. So some of the projects that are ongoing in our lab, there's the midline crossing of these Mouthner cell homologs. These are descending neurons that will cross the midline and go on to synapse onto other neurons in the motor ganglion, thought to be responsible for a sort of escape response now in response not to light, but rather perhaps peripheral mechanosensory stimuli. We're also looking at delamination and migration of dorsal root ganglion neuron homologs, the proposed homologs. These are cells that are specified at the very tip of the tail, and they will migrate towards the middle of the tail at the same time, forming these sort of bipolar processes that connect peripheral sensory cells to the central nervous system. Today, what I want to focus on is actually another story that sort of shows you that even though this nervous system is already, very small, in some species this seems to be getting even smaller. There are actually some species that are starting to lose this very small larval nervous system. In these species, as you'll see, this is mostly because they don't actually have a need to swim anymore as larvae. So, just this is just showing you a family tree of a particular branch of the tunicates. So obviously, there are way more tunicate species, but this is focusing on this large family, the modulates. And what's interesting about this family, as adults, they're sort of living on the bottom of the ocean for the most part. Most of these species are sort of living on sand flats and mud flats. And as a result, first of all, their larvae are not settling on the dark underside of things, right? So there's already less selective pressure in order for you to have some kind of specific behavior that's going to take a larva and have it swim towards a very specific spot. No, they sort of spawn. These eggs go up in the water column. They get fertilized. Maybe some tidal changes can disperse them, but at the end they fall to the bottom of the ocean, and that's where they live as adults. And this seems to have relaxed the need for an actual swimming dispersal phase. And so you actually have a lot of these species that have independently lost the swimming larval phase. So this is sort of showing you the distribution of either swimming larvae or non-swimming larvae. And these non-swimming larvae, as you'll see, they really have lost most of the structures that are required for swimming. And you see that even though you have a lot of these that do not have swimming larvae, the ancestral condition is a swimming larva. It's, this is an ancient chordate body plan, and we come from this sort of same body plan. And you see very clearly that there are certain cases where very closely related species are either swimming or not swimming, showing you that this evolutionary loss of swimming happened multiple times independently in different branches of this family. So this is a really interesting thing, not only from an evolutionary perspective, but also for us who are interested in the development of the nervous system, right? If something is, really important for larval swimming, for the nervous system, for that neural control of larval swimming, it's going to, you're going to maintain it, right? Through strong selective pressure to maintain that. But as soon as you lose that, that pressure to have a swimming larva, you're going to, the thought is that you're going to quickly lose, these things that you don't need. So by comparing what you've lost in these non-swimming larvae, you can, backtrack and identify what might actually be important for larval swimming. So we're sort of coming at it from, the opposite direction. This is just kind of showing you that these tunicates larvae, what will happen typically is that the larvae will attach to a substrate and undergo this process of metamorphosis where they quickly reabsorb that tail and now metamorphose into a sessile adult. And this whole process seems to be in a lot of these species, there seems to be sort of an ongoing evolutionary loss of this biphasic development. So you're going from sort of this indirect adult development to a more direct development. You have an egg, and you get embryogenesis, but then what hatches out of this egg is not really a swimming larva. It's rather something that's almost ready to become an adult. So a little bit more direct development. Yet these adults, right? They're normal, right? So they've just lost this larval phase here, but as adults they look, indistinguishable from those species that have a swimming larvae. So these are two adults from two different species. This one has a swimming larva. This one doesn't, but the adults are mostly the same, right? So this is a specific loss of specific structures in a specific phase of the life cycle, not a complete loss of the nervous system. This is a very selective loss of the nervous system development in the larval phase, right? So we're kind of interested in this question. So this is sort of comparing the larvae of these two species. Unfortunately, they were named by separate people at separate times, but just coincidentally now it's become very confusing. I hope to talk about mostly this species as a non-swimming or a non-swimmer, and this species, Oculata, as a swimming or swimmer species in order to avoid most confusion. You can already see that these two larvae are very different. The swimming larvae has that notochord, it has the muscles, and presumably it has this otolith. This is a pigmented sort of a heavy set, a cell that's heavy enough in order for it to detect sort of the sort of deflections as the larva changes its position, right? Similar to the otoliths in our own inner ear. Now, the non-swimming species has lost all these very obvious features. It's got muscle cells, but they're not differentiated. They don't form. It's got notochord cells, but these don't form a nice rod, a hydrostatic skeleton, and it's lost its pigment spot, meaning it doesn't really seem to have a functional otolith. These are just the obvious structures that have been lost, but we were really interested in understanding, what about the nervous system, right? You can't really see the nervous system just looking at it, from this, but what's actually happening to the larval nervous system? If it doesn't need to swim anymore, does it actually still need motor neurons? Does it need photoreceptors? Does it need any of these other components that are, for the most part, used to modulate swimming behavior typically? So that was the question that we asked. With has there been evolutionary loss of the larval central nervous system? And if so, what are the cellular and genetic basis for these these losses, right? What genes might have been lost, or have genes been lost, or is it more that certain genes are not expressed in the larva but they are in the adult? And what are the actual changes? What are the mutations that have occurred that can sort of tell us what's actually happening during this process of evolutionary loss? So we focused on a particular part of the tunicate larval nervous system called the motor ganglion. And this motor ganglion has been proposed to be homologous to the hindbrain / spinal cord. Not a clear distinction here because some of these neurons might be more closely related to neurons that come from the hindbrain in invertebrates, and others might be more closely related to neurons in the spinal cord. But they're all sort of scrunched up here in this small area here just at the base of the tail. So right posterior to the brain, at the base of the tail, it receives inputs, sensory inputs that are relayed from sensory cells either in the periphery or here in the otolith organ or the photoreceptor complex. And the connectome sort of shows that these cells are interconnected to form a network of cells that are, really sort of ultimately feeding onto two major pairs of motor neurons. And these are the cells that innervate the tail muscles, controlling the alternated left-right movements, but also sometimes the asymmetric movements that we see in the tortuous swimming escape responses. This is just this is really the cells that are there, right? And we're not talking about several cells like this, right? When we say that there's only one left motor neuron 1 and one right motor neuron 1, that's what we mean. So it really is just what is this? 1, 2, 3, 4, 5, 6, 7 pairs, left-right pairs of neurons that are controlling most of this swimming motor movement. And we focused on this not only because it's probably it's the best candidate for having been lost in a situation where the larva doesn't swim anymore, but also happens to be the neurons that we understand the most as far as their development goes. So we've previously shown that most of these cells actually come from a single cell in the 64-cell stage embryo. So on the right side, it'll give rise to the neurons on the right side. On the left side, it'll give rise to the same cells on the left side. But this is the cell lineage that can explain how these neurons are specified. And we know more or less the major events that are happening, signaling events that are specifying these cells, gene expression patterns, which transcription factors, which regulatory genes are important for determining the specification and differentiation of these unique neuron types in this. So having this information all coming from a swimming species, in this case Ciona, we went in to look at what's happening in one of the species that have lost the swimming larval phase, that is Mogila colta. And when we did in situ hybridization, so we're trying to detect the spatial expression patterns of various regulatory genes that seem to be specific for very, a determined cell type in this motor ganglion, we found that actually these cells seem to be there, right? Each of these individual cell types, motor neuron or motor ganglion neuron subtypes, seems to be there expressing the regulatory genes that they're supposed to, right? So these M cell homologs, the DDNs, they express this DMBX transcription factor in the in the swimming species, but in the non-swimming species, they're there as we were surprised to see that they were there. We expect maybe they're not specified or they're lost, but the cells seem to be there. Same thing for these motor neurons, so expressing this motor neuron regulatory gene called MNX. We see both pairs of motor neurons still there. This other transcription factor is expressed specifically in motor neuron 1, and so we saw that, yeah, there's a pair there. And then the two interneuron types, the two major interneuron types of the motor ganglion are there expressing this, this interneuron regulatory gene. So these are all actually homologs of transcription factors that are very important for our own, hindbrain and spinal cord motor neurons and spinal and interneurons. So these are not only had they been, shown that in the swimming species they seem to be carrying out very similar functions to what's happening in our own nervous system development, but now here we're showing that in the non-swimming species that they're still there. These important regulatory genes are still expressed in the right cells at the right time. So is there something else that we could tell, what else is happening downstream of this? Well, we also looked at certain other genes that were not, that might not be specific to one neuron or the other, but more generally involved in neural differentiation, right? Genes that, regulatory genes that are important for, after the neuron is specified for it to become actually differentiated as a fully functional neuron. And one gene that we looked at, very conserved, very important gene, is EBF. This encodes a transcription factor that's important for nervous system development. And we saw that, even though we could detect expression in other cells that were not related to the nervous system, so these are actually oral muscle progenitors, they will give rise in the adult to the oral mussels, and so they strongly express EBF in these non-swimming larvae. We notice very faint expression by RNA and CG hybridization of EBF in the neurons of the motor ganglion, and also sometimes in the brain, especially when compared to larvae of swimming species. So this is another Mogula species, but we had previously characterized expression in this species, and this is what you normally expect from this important neural differentiation factor, EBF expression, strong expression in these motor ganglion neurons. But of course, this is, so far this is just sort of a qualitative approach. It's not really a quantitative approach. But what exactly, is EBF? EBF stands for, well, actually, EBF is a part of this large family of transcription factor, the sort of Collier-Ulf EBF family, also known as CO. The orthologs, or homologs of these genes in many species are very important for neurodifferentiation, including in us, right? We had previously shown that EBF is critical for motor neuron differentiation. So in sort of these wild-type larvae, you can see a motor neuron, you can label it with a transgene, and if you knock out EBF using CRISPR, specifically in the territories that will give rise to the nervous system, you completely lose motor neuron specification and differentiation. And this has been shown to be conserved, since then, also in invertebrates, but also going all the way to C. elegans as well. So in nematodes, the EBF or CO ortholog 3 is a very important regulator of motor neuron differentiation. In humans, actually, one of the EBF genes has been recently found to be mutations in the EBF3 gene have been found to be actually quite a frequent cause of neurodevelopmental disorders. So these 3 papers back to back show that in up to 1 in 1,000 individuals, mutations in EBF3 explain previously unexplained neurodevelopmental disorders. These transcription factors are expressed in various different types of neurons throughout the central nervous system, presumably controlling the expression of various genes that are important for neural development, neural function as well. So this is a highly important, highly conserved family of genes that are important for neural development. And it seems to be that in these non-swimming species, the expression, at least qualitatively speaking, using this technique of in situ hybridization, didn't seem to be that high. But as I alluded to before, can we get a better quantification of that? Is EBF expression actually reduced in these motor neurons? And to answer that, we take advantage of the fact that these non-swimming and swimming species, the occulta and oculata, they actually occur in the same place, at least in this one place, in Roscoff, in the north of France, in Brittany. And taking advantage also of the fact that they actually form these little hybrid embryos that have a very, sort of a partially rescued tail, perhaps a partially rescued nervous system. These hybrid embryos don't actually, give rise to a functional or fertile adult, but they undergo development. The compatibilities are too great in order for this to actually, go throughout development. But you can, at the very least, analyze what's happening to the development in these hybrids. So taking advantage of this unique situation, in collaboration with Professors Billy Swala and Titus Brown at University of Washington and UC Davis, this project aimed at sequencing, using RNA-seq sequencing, quantifying gene expression changes in the 2 species plus their interspecific hybrid at 3 developmental time points. This is largely analyzed by Elijah Lope, then a graduate student with Titus Brown, but now a postdoc in our lab here at Georgia Tech. And so we've been able to continue this analysis, trying to understand what's exactly happening to the nervous system in this case. So what we expect, right, with using this technique of RNA-seq is that you're able to sort of quantify the extent of gene expression on a gene per gene basis. So if you have 2 genes, say gene X and gene Y, and you can actually see that, okay, maybe gene X and Y are expressed at similar levels in the swimming species. But in the non-swimming species, you're able to compare those levels and say, okay, it looks like there is a specific loss of expression of gene Y in the species. Furthermore, if you're sequencing the hybrid, you can also see if what are the dynamics in the hybrid as well. This allows us to answer another question that is more interesting from an evolutionary standpoint. What is the basis for the differences between the swimming and the non-swimming? These differences could be due to mutations in, say, the promoter, right? These would be called changes in Cis. So if you have different levels of expression, coming off of this one gene in the two species, in the hybrid, those differences should still be, the same, right? You should still be able to measure, a measurable difference between the alleles that are inherited either from the swimming parent or the non-swimming parent. The non-swimming parent isn't going to be magically fixed, right? Its promoter still has those sequences that, if those sequences are faulty, that expression is not going to be rescued. And you can distinguish that between changes in trans, meaning the trans-regulatory environment of the embryo. So if, let's say, the reason why in the non-swimming species this gene is not being expressed is because some transcription factor that's important for it has been lost from the species. Then if you make the hybrid, those differences are normalized, right? So you've got, you're making a hybrid between the two species. You've got maybe, in the best case scenario, maybe half the amount of that crucial transcription factor, and maybe you'll see that those differences are sort of averaged out. And so by comparing the allele-specific expression, so comparing the expression levels of the parental alleles in the hybrid allows you to understand more closely what's actually happening at the the level of evolution and genetic changes. So when we looked at the RNA-seq data, we could see that in a specific, as development proceeds, the non-swimming species isn't really activating that many genes. Right? So it seems that there's not a lot of gene expression changes happening as that tail is supposed to grow and the development of the nervous system of development is supposed to proceed. If you compare that to the swimming species, so in the swimming species we see a lot of genes that are enriched specifically at the tail bud stage. So anything above 0 here in these charts are genes that are going up during development. So more gene expression as development proceeds. And so this is kind of what you would expect, right? If you expect that this sort of non-swimming condition is a result of sort of an arrested development of sorts, you would expect to lose that significant gene expression that you typically see in the swimming species. So when we looked at specifically the transcription factors that we think are important for motor neuron differentiation, starting with neurogenin and one-cut and EBF. So these are all transcription factors that have been shown in other systems to sort of function as a regulatory cascade. We could tell that these genes, neurogenin and one-cut, they were still being upregulated at statistically significant levels in the non-swimming species, but EBF was not. So there seemed to be, at the level of when we were looking at this, the levels of RNA, that there was not a statistically significant increase in EBF expression in the non-swimming species at this critical time when the nervous system is developing at 6 hours. Furthermore, if you look at the allele-specific expression in the hybrid at that time, you see that neurogenin, there was no statistically significant difference between the two parental alleles, but there was for these other two genes, one-cut and EBF. So there seemed to be actual mutations in the promoters of these genes that seem to be reducing expression, maybe a little bit in the case of one-cut, but definitely a lot in the case of EBF. This is just the data. This data shown in a different manner. So this is sort of the effective read count. So this is just the number of reads that were sequenced, coming from this the swimming parent allele and the non-swimming parent allele in the hybrid, and there was a big difference. This is not just a generalized difference between the alleles of all the genes. So this is the plot, is a distribution of allele-specific expression of all the genes. Majority of genes actually showing no statistically significant difference between the parent alleles, right? It's either one or the other. Maybe sometimes a little bit one or the other, but not statistically significant. And in fact, the majority of statistically significant allele-specific differences actually is in favor of the non-swimming species. Probably has to do with the fact that these are we fertilize the non-swimming eggs with the swimming sperm, so a lot of this is maternal expression. But just showing you that only a small number of these genes are actually showing greater expression like this. So this is not just a sort of biased because of the experimental condition. We do believe that these are actual significant allele-specific expression levels that we can measure in the hybrid using this technique. When we looked at the activity of the promoters between of these two different species from that EBF gene, we had to do this in a species that we could work with here at Georgia Tech, and that would be Ciona. So we're not able to do these experiments yet in these actual Mogula species. But when we test these promoter sequences in Ciona, we could see that the swimming the EBF promoter from the swimming species was expressed in the central nervous system, in the motor ganglion, as well as these bipolar tail neurons, these sensory neurons. But the non-swimming species, its activity was specifically reduced in the motor ganglion. We don't see brain expression here, but when we looked at it more carefully, there was expression still in the brain and in these sensory neurons. So there's a specific loss, not just general loss of EBF expression, but specifically in the motor neurons of these larvae. So that would be sort of the quantification when we're looking at the expression, motor neuron expression of the swimming reporter. Seen usually in 50%. This is pretty good given that we're testing these constructs in a totally different species. But the non-swimming reporter, this loss, you see this almost no expression. Yet the expression in these other neurons, brain neurons, sensory neurons, is relatively well maintained. If you look at the sequences, you align them, you want to see what's happening. Well, we actually found a particular sequence that seemed to be kind of divergent. And this was interesting to us, mostly because this sequence here, called an E-box, is known to be bound by neurogenin, which is a transcription factor that's important for neural differentiation. So we already zoomed in on this one region. So zooming in on this one region, we decided to test whether these sequences, these differences in sequences, could explain the differential activity of these reporters. So we just swapped. We turned the swimming into sort of a more non-swimming sequence, and the non-swimming sequence into a more of a swimming sequence. And sure enough, when we take that swimming sequence and replace it with the non-swimming sequence, you get that same loss. So it seemed that sequence is really important in the swimming species promoter to maintain that expression. Unfortunately, we were not able to see the converse. We couldn't resurrect that non-swimming promoter, suggesting that throughout that whole promoter, there are probably several mutations that have accumulated over time. And this is kind of very important to stress. It's not that there's a single mutation and that's going to be the end of the story. No, if something is not needed, it's going to tend to first get lost, but then other additional deleterious mutations will continue to accumulate because now there's no difference, right? It doesn't matter if you've got one mutation or if you've got 10 mutations. That DNA is no longer needed in the species, presumably. So it'll tend to accumulate mutations until it's completely unrecognizable and deleterious. So what are the functional consequences of having less EBF in these cells? So if we think that at the level of neurogenin, at the very beginning of neural differentiation, there's no difference, but there does seem to be a species-specific difference at the level of EBF. What's the difference downstream of that? Well, so this is kind of converging with another point that we've found in Ciona, and I'll have to go through this relatively quickly. But in mammalian neuromuscular synapse maturation, there's a very important role for this extracellular protein that's secreted by motor neurons called agrin. You might remember this if you've learned about this in sort of a principles of neuroscience course. We learned about it in cellular molecular neuroscience and here at Georgia Tech, and also now neurodevelopment. But there's a specific role for agrin secreted by the motor neuron in order for it to generate sort of these neuromuscular synapse clusters. This is done through alternative splicing of agrin gene. And it was previously shown that this RNA binding protein, NOVA, is really important, specifically the neurons, to include these sort of small exons called Z exons, such that you knock out nova, you lose the inclusion, specifically of some of these exons. What are these exons actually encoding? They're encoding very short peptides that are important, at least as predicted from a.
It's not you.
It's not me.
It's somebody on at Emory
All right.
or dialing in.
Okay. Very important for interaction of agrin with its receptor in the muscles. The LRP receptor, this is sort of the crystal structure showing agrin and other related ligands, showing the very key residues here highlighted that are absolutely required for interaction with the receptor. So you've got this very important switch, an RNA-based switch. Alternative splicing results in these short peptides that are critical for neuron-specific agrin to induce neuromuscular synapse formation. In Ciona, we looked, it turns out that they also have these sort of Z exons, even though they don't encode the same amino acids, though they encode somewhat similar amino acids conserved here that would predict to sort of carry out the same function. So we've got something that at first glance seemed to be very specific to mammals, and now we've actually discovered that this is actually happening also in the tunicates. In retrospect, maybe not so surprising given that tunicates and vertebrates are so closely related. Agrin is expressed, and you start to see agrin inclusion, these Z exon inclusions in agrin as development proceeds. So same as you see in our nervous system. It's expressed in the motor ganglion. Nova is expressed in the motor ganglion. So you've got this sort of conserved circuit occurring in Ciona as well. I'm sorry I'm going through this really fast. I'm running out of time here. But you knock out nova in Ciona, and you start to lose the inclusion of these Z exons. And in mammalian cells, you can actually include those exons by overexpressing the Ciona nova. So these are just showing you that this nova is an RNA-binding protein expressed in the neurons, both in mammals and in tunicates. And in both systems, they're regulating the inclusion of these sort of agrin peptides that are absolutely, at least in mammals, critical for neuromuscular synapse formation. Is the same thing happening in tunicates? Yes. The answer is that when we do nova knockout in the motor neurons of the Ciona, you lose this typical clustering of these acetylcholine receptors at the neuromuscular synapse. And I didn't show you here, but the phenotype is copied when you also knock out the agrin Z exon. So you can just cut out those Z exons in agrin and get the same phenotype. You knock out the receptor in the muscle, not in the neurons, in the muscle, and you get the same phenotype, showing you that this whole pathway seems to be specific to chordates, right? Tunicates, vertebrates, and very important. So circling back, what's happening in these non-swimming species? Well, we looked at nova, and we found that it's one of these genes that actually is lost in the non-swimming species. So it's upregulated during development, as you would expect in the swimming species, but not in the non-swimming species. This is an in situ hybridization showing that it's expressed in other cells, but not in the nervous system. So you'd expect it to be expressed in the motor neurons. It's not. So its expression has been lost in the nervous system, but it doesn't actually show an allele-specific difference, suggesting that the differences come from what's upstream. In this case, we suspected it was EBF, right? So is EBF, sorry, that's just showing you the activity of nova and the lack of allele-specific expression. So is nova a transcriptional target? And the short answer is yes. When we do a knockout of EBF in Ciona, we completely lose nova expression in the nervous system. Now, this is showing you the percentage. Embryo showing in the control, you see nova GFP expression in the motor ganglion. You knock out EBF, you lose that. So it does seem that we've found a novel connection, a novel regulatory connection between EBF and nova in tunicates, in the tunicate motor neurons, and that this is probably very important. Well, we have shown that it's important for neuron-specific alternative splicing. And this could be conserved in vertebrates, right? We don't know. We're excited in following this up, collaborating with mammalian neurodevelopmental people, right? They know that EBF is important, especially in motor neurons, but they have no understanding of how nova is regulated in the mammalian spinal cord. They have no idea whether EBF regulates nova. So this is something that we expect might be highly conserved as well. So the conclusions here, larval motor neurons seem to be specified in the non-swimming species, but they're lost. The differentiation seems to be impaired, probably due to lost expression of this EBF. And we found that these differences occur primarily because of mutations in the promoters of EBF. And there does seem to be a link between NOVA expression and EBF function. So we predict that the loss of EBF would result in loss of NOVA, which would lead to downstream losses in neuromuscular synapse formation and possibly many other neural functions. So with that, I'd like to thank my lab. These are the core people in my lab, and not shown some of the undergraduates, but I'll be happy to take any questions if there's some time. Right.
And they work the same way in both. In other words, what happens? Are those motor neurons also located in the same motor ganglion?
No. So it seems that the motor ganglion itself doesn't make the transition to adulthood, and you have a more anterior structure that would be really more closely related to our hindbrain that would have these cranial nerves that are innervating those siphon muscles. Yeah. It's a different lineage. Yep. Yeah, and possibly very different promoters, regulating the expression of the same genes, probably though. Yeah.
So
yeah.
Really cool the manipulations you can do. I'm curious. You posed at the beginning that there were probably multiple losses of swimming, although the phylogeny was a little unclear how many there were. Right.
Mm-hmm,
but I was curious how much you thought this study served across different transitions, and maybe as a bigger question, if this is evidence for sort of parts of the neurodevelopment sequence that might be more or less modular.
Yeah, that's a great question. There have been various different instances of loss, and those are only the ones that we know about. There are hundreds of species that people don't even know if they have a swimming or a non-swimming larvae. There are some cases where it does seem that that this has happened maybe longer ago, or at least it's been more advanced. So at least in our species, we have some evidence of some vestigial, muscle or vestigial neurons. In some other species, it seems that maybe not even the vestiges are there. So it could be that they've been further lost. But in the specific case that we've looked at, the EBF, promoter, it does seem that there's a very modular organization to its expression, at least separating the expression in this motor ganglion as opposed to expression in other neurons. So we expect that it can lose the expression in the motor ganglion because that's a larval specific structure. But as it was pointed out, they have motor neurons in the adult they need to express EBF and agrin and NOVA and agrin and all these other components in the adult as well. But that's taken care of by different regulatory modules that are in different parts of that sort of promoter region. That's what seems to be happening. Yeah.
So you think like the sensory and the CNS parts might not be lost as easily because they might be integrated more into it?
Well, right now we're following up on understanding what else has been lost. So we're very interested in this whole photoreceptor. Has light sensing been lost? Maybe not, because the adult also senses light, but for different purposes, right? Yeah.
I'm curious about the Roscoff Brittany coexistence of these two species. So if you're losing a lot of the neurons in the larvae that actually allow you to escape predators or actually find your place to settle down, how did you end up having the evolution of a
the evolution of the loss, or the evolution of the loss?
Yeah, because one would imagine that the predators and everything are the same. So how is that species?
Right. So I think it has to do with the fact that this whole family is primarily mud and sand flat dwelling. You don't see these losses in these other families that all their species are attaching, to substrates and swimming around and stuff. So it does seem that loss is has occurred primarily in this family that their eggs are get buried in the sand, and presumably there's no swimming predator there. I mean, they might have to evolve other defenses that are not to do with escape responses. But there are, but yeah, there does seem to be evidence that also it has to do with tidal changes. So a lot of the species near the equator, where the tidal changes aren't as dramatic, they still need some swimming ability. Whereas at higher latitudes, it does seem that the dispersal role is carried out by these very dramatic tidal changes. The eggs go out and then they come back in, but maybe in a different inlet or something like that. So the geography and geology is going to be very important as well. All right.
Thank you, Bruno.